Okay, so let's talk about ABO blood group genetics. So first of all, you have to know that ABO blood group is actually an example of multiple alleles. So what is actually multiple allele? Multiple allele is simply means that if you can say form of genes in which you have more than 2 alleles, you can say they are called as multiple alleles. So in some genes may have like as many as 300 alleles, but in ABO blood group system you don't have that many alleles, but we will discuss about these alleles here. So ABO blood group system is an actual example of codominance. So what are the alleles that ABO blood group system have? There are 3 types of alleles. It is small I, then IA and IB. IA and IB, both of these are dominant, while small I is actually recessive. So capital IA, capital IB, both of these are dominant and small I is recessive. So when you do the cross, then you will have to know that there are 3 alleles, but you have actually 4 type of phenotypes that could be made. How these phenotypes are made? Let's go there. So first of all, if you have genotype, that is capital IA or capital IA or small I. If you have small I and capital IA, obviously capital IA is dominant on small I, then the blood group would be A. If you have a genotype that contains capital IB or capital IB and small I, then the blood group would be B. But if you have a genotype that contains capital IA and capital IB, then both of these would be shown and the blood group would be AB. AB contains both alleles IA and IB, but only if you have small I and small I, you will have blood group O. That's why O blood group is really rare. O group is only possible when you have homozygous recessive. You know that homozygous recessive is very rare. It have really low probability to come in your progeny. So that's why O blood group, if you have a O blood group, you are rare, and if you have O negative, then you are more rare. We will talk about positive and negative later, but you have to know about the blood group first, then we will move on to positive and negative ones. So first of all, you have to know that ABO blood group system, this system was actually discovered by K. Landsteiner. That was the scientist who discovered ABO blood group system. So he discovered that the ABO blood group actually contained a polymorphic gene, that was I, and it had 3 alleles. This gene was capital IA, capital IB and small I. So these were the 3 alleles, and these 3 alleles actually had 4 phenotypic possibilities. Either you have A blood group or B blood group or AB or O. So the genotype are in front of you. If both are dominant and homozygous or heterozygous, homozygous and heterozygous, and you have heterozygous. So if the heterozygous is AB, then it will be AB, and if it is homozygous recessive, only in this condition it would be O. So let's talk about antigens and antibodies. What are antigens and what are antibodies? So antigens are actually small proteins. For example, this is a red blood cell in front of you, and this red blood cell actually have a small protein on it, and this small protein is called as antigen, and this antigen decide if your blood group is going to be A or B or O, right? If the antigen is A, then the blood group would be A. If the antigen is B, then the blood group is going to be B. If there is no antigen at all, then the blood group would be O. So the presence or absence of antigen and the type of antigen will decide your blood group. And what about antibodies? So antibodies are big macrophage structure. They are really big, like this, plate-like structures, and these antibodies are actually your army in your body. The army in your body, they are actually protectors of your body. They are part of immune system. If any foreign particle or any enemy comes into your body, antibodies will go there and kill that foreign particle. So why are we discussing about antibodies in blood group system? Well, because antibodies have specific receptors that have specific antigens to receive. For example, the antibodies in your blood group only receives the blood that is same as you. If you have a blood group A, then you will have antigen A, but the antibody will be B. Why is that? Because if antigen B containing blood come into your body, then that is not a part of your body. So the anti-B antibodies will kill those antigens, right? If you have antigen B in your blood, then you will have anti-A antibodies, because if antigen A containing blood comes into your body, then antibodies will kill the antigen A containing blood. So antigen B will have antibodies A. So antigen A containing blood don't come into antigen B containing blood. So B blood group will never receive A blood group. The reason is because antigen B containing blood group will have anti-A antibodies. If we talk about AB, because blood group AB has both A and B antigens, so that's why it will not have anti-A or anti-B antibodies. The reason is that because if you have both A and B antigens, then the antibody will kill either A or B. So to protect your body from its own antibodies, you don't have to get an antibodies that should be neither anti-A neither anti-B. That's why you can say AB can receive any kind of blood. You know that A can only receive A type of blood, while B can receive only B type of blood, while AB can receive A blood group and also B blood group and AB blood group and also O blood group, because it does not have antibodies at all. And though if it does not have any anti-A or anti-B antibodies, then those antibodies will not kill anyone. Obviously they will have antibodies, but not anti-A or anti-B antibodies. You have to know that there are types of antibodies in your body. AB does not have any antibody at all. No, they have antibodies, but their antibodies will have different functions, but they will not kill antigen A or antigen B. Okay. If we talk about O blood group system, then the O blood group system is actually opposite. O blood group system, you know that have recessive I and recessive I. So it will not have neither A antigen or B antigen, and it will actually have both anti-A and anti-B antibodies. It is actually opposite of AB blood group system. That's why you can say that the O blood group can only receive from O blood group, but you know that it does not contain any antigen. If it does not contain any antigen, then it can go to anybody at all, right? If it does not have any antigen, then antibody cannot kill it. You are understanding my point? If there is no antigen, then how antibody can kill it? If antibody cannot even recognize the antigen, because antibody will see that, oh, this is antibody B, I have to kill it. This is antibody, or if this is antigen A, I have to kill it. But if there is no antigen at all, how will antibody will recognize that cell? And if it cannot recognize it, then antibody cannot kill that cell. That's why O blood group is actually universal donor. So blood group A can receive any blood group, they are called as universal recipient, and blood group O are universal donor, they can donate to any blood group possible. Okay, so if there is a chart right in front of you, as you can see here, if there is a father that has blood group A and there is a mother that has blood group A, then they can have either blood group A or O. Why is that? Let me write it down in front of you. Father and mother. Father can have either capital IA or capital IA or small I, right? And mother can also have capital IA or capital IA and small I, right? So both of these conditions are possible. If both of them have capital A, then all of their kids would be A blood group. But if father have small I or if mother have small I, then there is a probability that their one of these children will have a O blood group. Let's say that mother has the small I antigen, or sorry, small I allele, then the gametes of father are small capital IA, and mother has capital IA and small I. We have taken this example. If we make their cross, then you will have capital IA, then capital IA and small I, right? Then again you will have capital IA and capital IA and small I. So you got both of these that were heterozygous and you got two dominant, and when they cross again you will get capital IA and small I. So there is a chance that they can have small I children in the future, right? So you have to know how to make these kind of cross, because these kind of cross to make are really important. Now you understood how to make a cross and how to know the blood group and how there is a probability in these things. I think I have made blood group system really clear. The next thing is that you have to practice again and again. So this is a table that is right in front of you. Practice it as much as you can. This is really important. What if the blood group is A, blood group is B, blood group O, then this is table right in front of you. What is the probability of child's blood type? This type of table could be in your IMAT exam. So I hope you get to know how to make pedigree charts. Then making this type of table would be really easy for you. The first thing is that you have to know how to make a pedigree chart, right? If you know how to make a pedigree chart, then these kind of MCQs would be really easy for you. Did you get my point? So next thing is about, if you haven't we haven't studied about the blood group system. So first of all, let's talk about how positive and negative blood groups are actually made. So this is actually called as RH blood group system. It was again discovered by Kale and Steiner, that we discussed about. Again, why is it called RH blood group system? Well, RH came from rhesus monkey. The experiment was done first on a rhesus monkey, so that's why it is called as RH blood group system. So what is RH blood group system? So there is actually type of alleles, 3 types of allele that are really important in deciding RH blood group system. There are 3 types of genes. The 3 genes are actually C, D and E. The alleles for gene, you can say the alleles of gene D are actually present on one locus, and the other 2 genes are gene C and gene E. Both of these genes are actually present on one locus. You know that for you have studied until now that each allele will have one locus and the other allele will have another locus, but C and E are the alleles that are, both of these alleles are present on one locus. So alleles of C and E are on one locus and G on one locus. D is actually the important one here. So D allele is like the one that determines your blood group is going to be either positive or negative. For example, let's write down RH positive, when you will have RH positive and when you will have RH negative blood group system. Let's make a table here. So if you have an RH factor, if the RH factor is present, then the blood group would be positive. If the RH factor is not present, then the blood group would be negative. What would be the genotypes? The RH factor is actually present on the D alleles, right? The allele D actually inhibits the formation of RH factor. If the genotype is capital D small d, or you can say capital D, then the blood group would be positive. If you have small d and small d, then the blood group would be negative, right? So this is the one thing that you have to keep in mind. Okay, so this was about blood group system. So you have to keep in mind what and how blood group system is actually made. I think we can add more things here. The next thing that we can add here is whether which can donate to whom. So first of all, you need to know that Rh positive can donate to positive blood groups, while it can receive from positive and negative. So it can donate to positive, and it can receive from positive and negative both, right? If we talk about negative blood group system, then negative blood group system can donate to all of these, like it can donate to positive, it can donate to negative, right? But it can only receive from negative blood group. So this was the things that you have to keep in mind for blood group systems. The arrow is telling you about receiving, this arrow is telling you about donation, this arrow is also about donation, this arrow is about receiving. So Rh positive can donate to positive and receive from positive and negative, while negative can donate to positive and negative too, but can only receive from negative one. So these were the things that you have to keep in mind about Rh blood group system. So this was about blood group systems that were related to your IMAT exam. You have to be really careful about what could be asked from this blood group genetics. Okay, so the blood group genetics, your topic is complete. You don't have to study anything else after we have discussed until now. This is the most that could be asked in your exam. The next thing is going to be about sex linked inheritances. So what is sex linked inheritance? You can take a break again, you can come back, and we will discuss about sex linked inheritance. Okay, so let's talk about sex linked inheritance. So you know that there are types of chromosomes. You know that there are 23 pairs of chromosomes, and out of 23, one pair is called as sex chromosomes and the remaining 22 pairs are called as autosomes, right? So the one pair, that the sex chromosomes, actually defines what is your going to be biological sex. Either you are going to be male or female is decided by this pair of chromosomes. If you have X and X chromosome, then it is going to be a female, and if it has X and Y chromosome, then it is going to be a male. So the thing is that who determines the sex of offspring? Then if you can say that, then it will be father, because Y chromosome is in the father. So if female is giving X chromosomes, then either X or Y will come, depends on the father. So if you are a boy, then the Y chromosome that you have actually came from your father, and if you are a female, then one X is from your mother and the other X is from your father. So either you are going to be male or female actually depends on your father, not on your mother, because the X and the second Y and second X actually comes from the male partner, right? So when we are talking about inheritances, then we will talk about the inheritances that comes from X chromosome and then from Y chromosomes. So first of all, if we talk about X chromosomes, then the inherit characters that are from X chromosome either could be dominant or either could be recessive. So first of all, let's talk about X-linked recessive inheritance. So the characters that are inherited from recessive traits are called as X-linked recessive inheritance. For example, let's make a pedigree chart. First of all, imagine this is your father, right? And the father is normal, and this is the mother, and the mother is containing recessive trait. Let's say the recessive trait is small h and normal is capital H. This is also normal. So this is the father and this is mother. And if we make the pedigree chart here, then you will get X, Y, X and X, right? So one chromosome of mother is actually the recessive X chromosome and other is dominant. Same is for father, it contains only one X chromosome that is dominant. We are taking this as an example, okay? So let's make the pedigree chart. So as you can see here, first of all, if we make the first column, then the first column will have X and X. One of them have small h and the other is capital H. If you see here, then this is first of all, what is it? It is a female, but it is normal. Why is that? Because there is a capital H. Capital H is dominant on small h, so this would be a normal female. Let's move to the next column, that is X capital H, X capital H. This is also a normal female. And if we draw here, then you will have X small h and Y. As you can see here, this offspring actually has X, that is small h, and Y does not have that inheritance, because we are talking about X chromosomes. This inheritance, this trait is on X chromosome. So the trait that is on X chromosome is actually recessive one. So that means this trait will be shown in this individual. That means this boy, this male will have that trait. And if we talk about here, that it would be X and Y, and this X is actually capital H. So in this situation here, you can say, first of all, the trait is shown in this boy. This boy has shown the character, while the female also has one X, that is small h. It has recessive X chromosome, but it also have dominant X chromosome. That's why this female is containing the recessive allele, but it is a carrier. You can say what it is. It is a carrier of X chromosome, but it does not show the trait. While this boy only had one X chromosome, and that X chromosome had small h. That's why it is the boy that had shown the character. So you can say that X-linked inheritance is more common in males. Why is that? Because as you can see, in male you need only one X to have small h, then they will show the character, while in female both X should have the recessive trait for it to be the expression, right? So to express that trait, both X chromosomes should have the recessive trait, while in males only one X can show the trait. That's why you can say the X-linked recessive trait is common in males. Simple, right? So that's why you can say X-linked recessive are more common in males. The reason is because males contains only one X chromosome, and if that X chromosome has capital H, then it will be normal, but if it has small h, then it will show the character. Most of the X-linked recessive inheritance also contains diseases. So X-linked recessive diseases are very common. You have to memorize their names. The first example is hemophilia. Hemophilia is an X-linked recessive disease. The other example is color blindness. If there is someone that is color blind, then that is also a X-linked recessive disease. Then you have dystrophy. This is also a X-linked recessive disease. And then the last example is testicular feminization. These are most important examples of X-linked recessive traits, feminization. Okay, so these are the examples that you have to keep in mind when you are memorizing X-linked recessive traits. So there are examples right in front of you that are for X-linked recessive traits. As you can see here, this is an unaffected father. It has normal X chromosome and Y chromosome, while the female has mutated one chromosome, that this X1 is actually mutated and the other X is normal. So when the pedigree analysis was made, you can see here, just like in our chart, there was one affected son, just like in our chart here, right? And there was a carrier daughter like this one. So there was a affected son and there was a carrier daughter that had the trait of recessive one, but it was not shown, it was carrier. And there was an unaffected son and unaffected daughter. Both of them didn't had any X-linked recessive inheritance. Also, if we have affected father and unaffected mother, then you will not be affected, none of the sons would be affected. Why is that? Because both of these sons will get their X chromosome from their mother, right? And they are getting the Y chromosomes from their father. So both of these sons would be unaffected, but both of these daughters would be carriers. And when both of these daughters are carriers, then you can say that if the carrier mother marries a normal male, then it will have a affected son. So the X-linked recessive, you can say, moves in a zigzag manner, or you can say it does not go directly from father to son. Why is that? You can see here there was an affected father, and in that affected father passed down the X chromosomes to the daughter, but the male actually got the X chromosome from their mother. The sons actually got Y chromosome from father and X chromosome from mother. So if they hadn't gotten the X chromosome from father, so they will be unaffected. So the father actually produced two carrier daughters, and when those carrier daughters marries a normal male, then then after that the son would be affected. So you can say that the disease or the X-linked recessive traits are transferred from grandfather to the grandson, not directly from father to son, but actually from grandfather to grandson. So it is a zigzag manner inheritance that does not pass directly from father to son, but from grandfather to grandson. So that is a really important concept that you have to know, that X-linked recessive trait travel in a zigzag manner in the family history. So that was about X-linked recessive inheritance and its examples. As you can see here, these are the phenotypes and their notations. If both X are capital H, then it would be normal, and if it has one XH, then it would be a carrier. And if both of these X are small h, both of these are recessive, then that is the only case a female will be affected. So for a female to be affected, the chances are really low, right? So for father and mother, if for both father and mother are affected, or you can say both father and mother contains X-linked recessive trait, then in that case the female would be affected, otherwise the female would always be a carrier. And for a male, being affected is really easy for a male, because only X need to be recessive, it contains only one X, and if that one X had that X-linked recessive trait, then it would be affected. Either it could be hemophilia, either it could be color blindness. So that's why hemophilia is more common in males and less common in females. Let's move on to our next one, that is about X-linked recessive trait appear more frequently in male. We have already discussed it. The cross about carrier mother and normal father, we have already discussed that. So this was all about X-linked recessive traits. Let's move on to our next topic, that is about X-linked dominant trait. So X-linked dominant trait is, you can say, much rare than X-linked recessive trait, and it can actually pass from father to all of his daughter, and like all of they receive the X chromosomes, but no son will be affected. So it directly goes from father to daughter and never from father to son. So X-linked dominant inheritance is more common in females. As you can see here, there was an affected father right here, and that affected father passed down the X chromosomes to the daughters. So that's why these are the affected daughters right here. You can make the pedigree chart again. I am like, this is your homework. Go to open up your notebook, and you should make this chart right now about how X-linked dominant inheritance goes directly from father to daughter. and then if there is an affected mother, then that is the only chance then that you can have an affected son, because a son gets the X chromosome from the mother and you will have one single affected daughter. So that's how X-linked dominant inheritance actually works. So that's was all about X-linked recessive and X-linked dominant traits. And the last thing is about Y-linked traits. So you can say that Y-linked traits are the traits that passes from father to the son, right? You know that Y chromosomes comes from the father to the son, so that's why Y is the maleness, right? So only males have Y chromosomes, so that's why Y-link are called as Holindric traits. What are they called as? They are called as Holindric traits. I think I have written it down somewhere, yeah, here. So Y-linked traits are called as Holindric traits. They pass from father to all their sons and never to the daughters, because daughter or a female never have a Y chromosomes. So Y chromosome, it is like very rare, but you can say Y chromosome called maleness, and if you have hairy chest and beard, then all of these characters are related to the Y chromosomes. Okay, so let's talk about some of the key identifying features. So for example, if you have given an MCQs, then these are the clues that can help you to identify either it is autosomal recessive, autosomal dominant, X-linked recessive or X-linked dominant. If both of the sexes, males and female, are affected equally, then you will know that this the disease is not X-linked recessive or Y-linked recessive. The disease is being passed from autosomal chromosome. Autosomal chromosomes are given to both males and females equally, right? So if both sexes are affected equally, then it is going to be a autosomal disease. And let's talk about what either it will be recessive or dominant. Well, it would be really easy. If the two parents are affected and all their children are affected, then it is autosomal. And if only one parent is affected and 50% of the children are affected, then it is a autosomal dominant disease. So these are the additional clues that you can have. The next thing is about X-linked recessive and X-linked dominant. You know that if more males are affected, then it is X-linked recessive, and if it is being passed from father to daughter directly, then it is X-linked dominant. Next thing is about Y-linked Holindric traits. Then you know that it goes from all males. If all males are affected, then it is a Y-linked Holindric trait. So take a screenshot of these additional clues and identifying features. They will help you a lot in your IMAT exam. The next thing is about these are the pedigree charts that are in front of you about the autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. So these are the shortcuts and like charts that are in front of you that you can take screenshot of. And this was all about dominance and their relationships, and I think we have discussed all of these in detail, and I have given you the shortcuts about the pedigree charts. So you just have to memorize them, practice them as much as you can, solve as many MCQs as you can. When you solve a lot of MCQs, then these concepts will be on your fingertips. The last thing is about genetic linkage and chromosomes. You already know about gene linkage. So what is gene linkage? Gene linkage is actually really simple. So it is actually a physical phenomena. Gene linkage is what? A physical phenomena. It is a physical relationship in which like all of the genes stay together in a link. That is simple called as gene linkage. So they combine together, inherit together as a unit. So it is called as gene linkage. And what about crossing over? So these are the two things that you have to be really careful about. So you know that there are actually 23 linkage groups in humans. Humans have 23 linkage groups. 23 linkage in humans. So humans have 23 linkage groups, and because of these linkage groups, you can say that Mendel's law does not follow. Why is that? Well, you know that for Mendel's law to follow, if you have two chromosomes RR and YY, then they have to separate out and form gametes like this. But if they don't separate out and they combine together with each other, then obviously the Mendel's law would not be able to follow at this point. So you can say that gene linkage caused the Mendel's laws of inheritance invalid. So gene linkage causes the Mendel's law to be invalid in certain conditions. So if there is a gene linkage, then Mendel's law would not be valid at that point. So you have to keep in mind that gene linkage is actually inversely proportional to the Mendel's law. Let me write it down again. Mendel's law would be invalid in gene linkage. So this is the point that you have to keep in mind. So gene linkage simply means gene pairs, or you can say homologous chromosomes, or you can say chromosomes in a gametes. So when gene linkage is formed, then Mendel's laws become invalid. If we talk about crossing over, you know that what happens in crossing over. In crossing over, you have already discussed in cell cycle chapter what happens in crossing over. In crossing over, you actually get genetic recombination. What is genetic recombination? What is crossing over? You have already discussed it, but I will discuss it for the one last time. If you have two chromosomes like this, one chromosome and other chromosomes, they will come together, they will form synapse with each other, and they will actually share arms with each other with non-homologous chromosomes. The non-homologous chromosomes will actually share or exchange parts of their DNA with each other. That is called as chromosomes. And after that, you can say that both of these chromosomes recombine their genes with each other. This is called as genetic recombination. When genetic recombination happens, then what happens is that you can say that there would be less gene linkage. There would be less gene linkage. And if the gene linkage is high, the crossing over would be less. So the concept here is the more gene linkage, the less crossing over. The less crossing over, more gene linkage. If you don't understand it, let me make it simpler for you. For example, if there are two chromosomes, like one chromosome is here and another chromosome is here, and if both of these chromosomes formed a gene linkage with their neighboring chromosomes, what will happen at this point? These chromosomes would not be able to come close to each other, right? Their distance will increase. When their distance increase and the gene linkage actually keep them together, and gene linkage will not let them to exchange their genetics, and if they can't exchange, their crossing over will not happen. So the more gene linkage is present, the less crossing over will happen. So that's why you can say that gene linkage is inversely proportional to crossing over. So for Mendel's laws to be valid, you need crossing over, you need genetic recombination. If you don't have genetic recombination, then Mendel's laws would be very difficult to show the phenotypes, right? So this is the thing that you have to know. If you want me to make it more simple, then let's make a flow chart so that you can remember it for like eternity. Okay, so let's make a flow chart. The first flow chart would be about linkage. Imagine if there is a linkage is present, and if linkage is not present. So there could be only two conditions, right? If the linkage is present, then what will happen? Then crossing over would be less, right? And if the gene linkage is not present, then crossing over would be more crossing over would happen, right? Because genes would not be held together and they would be able to exchange segments very easily. And if there is less crossing over, then you can say that you will have more parent types. The progeny would be just like parent types. And if there is more crossing over, then you can say that more recombination will be more, and you will have less parent types, like the children would be less like parents. So this is the thing that you have to keep in mind. If there is a gene linkage, there would be less crossing over, and the offsprings or the kids would look like just like parents. But if there is no gene linkage, then there would be more crossing over, and the offsprings would have more recombination, and there would be less likely to be like parents. So you know that the gene linkage in humans are 23. There are not so many gene linkage. That's why there is some crossing over in humans, and that's why you are not exactly like your parents. The reason is that because of gene linkage. So gene linkage actually prevents you to look exactly like your parents. So if there is less if there is less gene linkage, there would be more crossing over, and there would be more recombination, and you will look less like your parents. So that was the concept about crossing over and gene linkage. So this was about the concept about genetic linkage and crossing over. The last is about Hardy-Weinberg equilibrium. You can say that population genetics. So Hardy-Weinberg equation is a low yield topic in your IMAT exam. There would be application based questions. They wouldn't ask you about what is Hardy-Weinberg theorem, what are its conditions. What you have to know is that what is the equation of Hardy-Weinberg theorem and how to utilize it, right? So first of all, you have to memorize the equation, that is p square plus 2pq plus q square equals to 1. So what is p? P is actually the homozygous dominant, q is homozygous recessive, and pq is heterozygous frequency. So how many homozygous dominant alleles you have, how many homozygous recessive you have, and how many heterozygous you have. These are the 5 conditions that shouldn't be violated. These are the conditions that should be followed for Hardy-Weinberg equation, right? Without these 5 conditions, the equation would be invalid. So let's solve an MCQ that contains Hardy-Weinberg theorem. For example, they are saying that in a population 9% of individuals have cystic fibrosis. What is the carrier frequency? They have told you that this is the frequencies of the disease. So you can say that what is cystic fibrosis? Cystic fibrosis is actually because of homozygous recessive disease, right? So if it is 9%, then you can say q square is equal to 9%, or you can write it down as 0.09. So if q square is 0.09, then q would be 0.03, right? So if you know q, then you can find p. To find p, so actually p and q are actually equal to 1. You know that p plus q equals to 1 from this equation. So if you want to find p, then p would be equal to 1 minus q. So 1 minus what is q? Q is 0.03. Then you will get 0.7. You found p. So if you have q, then you can find p, and if you have p, then you can find q. So it is as simple as it is. Okay. So next thing is the thing that the question that they didn't mention was that cystic fibrosis is recessive. So the examiner in IMAT will obviously tell you if the disease is recessive or not. Oh sorry, it actually told, it was told in the question here. I didn't read it, sorry about that. So cystic fibrosis is autosomal recessive. From this statement, you know that the 9% is actually the q, right? So q was 0.03. From q you found p, and now you have q and p, then you can use it in the equation. So what is the equation? The equation is p square plus 2pq plus, and last thing is what is it? It is q square equals to 1, right? So what they have asked is, what they asked about what is the carrier frequency. So the carrier frequency would be 2pq. So whenever you have to find the carrier frequency, always go for 2pq, because carrier is the mixture of homozygous and heterozygous, right? So 2pq would be equal to, what is p? P is 0.7. What is q? Q is 0.3. Then the answer would be 0.42, or you can say that this is also 42%. So that's how you can use Hardy-Weinberg equation. So these kind of questions wouldn't be asked in your IMAT exam, because this is a low yield question. But you have to know that this topic is actually related to your IMAT syllabus. If you are thinking that you can skip this topic, I wouldn't recommend it to skip this topic. You have to know the definition about the Hardy-Weinberg theorem and definitely its equation. And right now we solved a question to know how to solve Hardy-Weinberg equation. These questions are really easy if you know the equation. P square plus 2pq plus q square is equal to 1. What is P? You know that P is homozygous dominant, Q is homozygous recessive, while PQ is heterozygous. Simple, right? So you will be given homozygous dominant or heterozygous dominant or homozygous recessive, and you have to find the other ones. So using this equation you can find any of these. This is like simple basic math, basic algebra. You don't have to be really worried about this topic. So I hope so you learned a lot of things from this chapter. I tried to try to remain as topic friendly as I can. I didn't get too deep this time because this chapter, or you can say these concepts, are very vast, and we can go really deep into these topics. We can solve a lot of questions for hours, but for today I think the lecture is already very long, so we will finish it here, and then in our future lectures we will solve a lot of questions, a lot of practice, and then you will be able to solve all the biology related concept questions really easily. So that's all from my side. Take care. Bye bye.